Hello. My name is Mr. Chipman. I am the AP Biology teacher at Murray High School in Murray, Kentucky, and this is Unit 5 review of AP Bio. Unit 5 is Heredity, not called Review. I don't know why I wrote the word review twice, but that's going to be just fine. We're going to talk about heredity in this video. In order to talk about heredity, we got to talk about this process called meiosis. Meiosis is not a cycle. Love this picture because it shows it is not a cycle. It is a process that has a product. It's different than mitosis. Mitosis is technically not a cycle either, but it comes at the end of a cycle and it produces something that begins the next rung of that cycle. When meiosis is over, or meiosis, as some people pronounce it, I'm going to call it meiosis. This is my video. But when meiosis is finished, you have 4 cells that are genetically different from one another. You begin with a diploid cell. Diploid or 2N cell. Diploid meaning that they have both copies of a chromosome, one from mom, one from dad. And at the end of that, those cells are haploid cells, meaning that they only have one of each chromosome pair. It's kind of like thinking about pairs of shoes. You have left and right, but at the end of meiosis, you only have left or right, and you have that for every single pair that you own. And there's 4, and they're genetically different. So that is what makes meiosis different. If you have that happening with the other individual as well in the production of gametes, then you have the potential for lots of variability, and that's why sexual reproduction creates much more variability than asexual reproduction. This is those terms, haploid, diploid. One copy of each chromosome. Haploid, N is typically the symbol that is used for that. And for diploid, you have 2 copies of each chromosome. 2N is the number or the symbol that is used for that. And what we call these pairs here are homologous pairs. They're homologous. They're not identical. Notice that they've done a good job in this picture showing you that they're not identical. They are homologous in that they have the same genes on them. They may not have the same versions of those genes. We'll talk about that in a moment. But they have the same genes. So mom gives you purple, dad gives you pink. Both this chromosome for both have the eye color gene, but one gives you blue, the other gives you brown. It's 2 different versions of that gene. So that's a homologous pair. A haploid only has one of each homologous pair. Moving on. Let's talk about the first parts of meiosis. Meiosis begins with interphase. Interphase is just a one-time thing. The important thing to remember for interphase is that this is when DNA is replicated. You cannot do this without DNA replication because you're cutting things in half. And so to think about it in the human cell, humans have 23 pairs of chromosomes. And so after meiosis, or after interphase, you're going to have 23 pairs twice. You could think of it as 46 pairs, but they're not different pairs. You've duplicated 23 pairs, and so you could say 92 total chromosomes. Because meiosis has 2 divisions, coming back up here, see 1 and then 2 divisions, you have 92 divided by 2, 46 divided by 2 again, you got 23, 4 times 23. And so that's how you have the 23 for human cells or whatever it would be for the other cells. And for meiosis 1, you have prophase, metaphase, anaphase, telophase. The important phases for meiosis 1 are prophase 1 and anaphase 1. And I think we'll get to that in a minute. If not, I'll come back to this slide. I don't quite remember what my slides look like. And that's the first division. At the end of telophase 1, you have cytokinesis, and then you start with meiosis 2. Not a whole lot to talk about here except for anaphase 2. We will make a big distinction between anaphase 1 and anaphase 2. Those are the important phases there. Prophase 1 has some other things that are going on that make it important, but we will talk about that later. We'll talk about that actually in just a moment. So this shows you the difference between mitosis, meiosis. Big deal. Mitosis is creation of 2 diploid daughter cells that are identical. Meiosis, 4 genetically different haploid cells. And actually, we're going to go back up here because we're going to talk about the difference between prophase 1 first and then anaphase 1 and anaphase 2, making the big differences. In prophase 1, you have something called crossing over that occurs. And what is crossing over? And so crossing over is when homologous pairs line up. And so if I were to, let's go back up here, and let's say that purple and pink duplicate. Well, I'm going to just demonstrate this on my screen because I can. I'm just going to have red and red. But let's say this is a homologous pair. Why do they look like this? Because they've duplicated, right? And so when this pair gets really close to this other pair, they're going to kind of form this little structure right here. And this is called a tetrad. And when those tetrads form, let's see that right there. Tetrad, why tetrad? Because the prefix tetra means four. And what can happen is crossing over can actually occur. And let me go down to my crossing over picture. So here's my homologous pairs. This is so they're aligning, so tetrads form. And crossing over is where they can actually swap sections of the chromosome because they're lined up. And this wouldn't do any danger to this chromosome because these two chromosomes have identical or homologous genes on them, the same genes, maybe different versions of that gene. And so crossing over creates some inherent variability to sexual reproduction as well. Another big difference, anaphase 1. During anaphase 1, homologous pairs separate. And so during anaphase 1, purple is going to separate from pink. And blue is going to separate from light blue, and green from light green. They're going to separate. And so after meiosis 1, or after anaphase 1, or meiosis 1, I guess, you have two haploid cells. Why are they haploid? Because they only have one copy of each chromosome. And this particular picture shows the phenomena of crossing over, right? Because you see all the little pieces in there. So it's actually helpful. And that is anaphase 1. Anaphase 2 is when sister chromatids separate. So what do I mean by that? So here's my chromosome, right? And in anaphase 2, sister chromatids separate so that each cell only gets one chromatid. And so the other phases are just similar to the cell cycle that we've learned, with the nuclear envelope disappearing and reappearing and the thalings lining up in the middle. But the big happenings in meiosis 1 and 2 are prophase 1, where tetrads form and crossing over occurs, and then anaphase 1, homologous pairs separate, or anaphase 2, sister chromatids separate. Moving on, let's talk about some places that variability can occur. Crossing over is one of those. We mentioned that. Just remember that homologous portions of homologous pairs cross over, and there's no damage to either one. This happens quite a bit. There's also independent assortment. What is this? This is one of Mendel's laws. Mendel said that individual traits assort themselves independent of one another. Well, Mendel didn't know much about chromosomes at the time, but we can amend that to understand that one chromosome is going to be inherited separate from another chromosome. And so what does that mean? Well, on this particular portion, we have Y is one gene, and it's found on big chromosome, or bigger chromosome, and R is found on the smaller. So R is a set of homologous pairs, and Y is a homologous pair. Well, when they separate, blue and red are going to separate, right? Big Y and little y are going to separate. Now, which way does big R go? Well, it could go with blue, or it could go with red. And so what this is showing you is it's showing you the possibilities. So it's showing you that big, it could go blue, blue, red, red, or it could be blue, red, red, blue. Well, how do we show this with like a Mendelian kind of cross or something? And so let's say we have an individual that, I don't like using Ys because it's too similar. Let's say we have an individual with this genotype, right? Well, we know that big A and little a are going to separate from one another because that is the law of segregation, which we didn't talk about, but we've talked about before in like an honors class or something like that. But the law of independent assortment says that big B could go with big A or with little a. And big B or little a could go with big B or little b. And so that individual here has the probability of producing any one of those combinations. Well, you take a human cell that has 23 pairs of chromosomes, and you can see that you're going to get into some pretty hefty arrangements all of a sudden. It's like 2 to the 23rd power different combinations that you can have, which is a fairly sizable number. So independent assortment is going to create variability as well. And then fertilization. How does it create variability? Well, for the male, the male is producing 4 different haploid cells. The female also produces one, but only one of those is going to be the egg cell. And so any one of these 4 could combine with that egg. And then you think, well, that's not very much, but any one of those 4 for the female could be picked to be the egg, or it's not a picking process. I'm not exactly sure how that works, but I do know that only one of those 4 haploid cells becomes the egg. And so there's a lot of variability here as well. There's a 1 in 16 chance that any one of those would combine with any one of these. And so that is a pretty significant variability when you combine that with independent assortment and with crossing over. There's a lot of variation that can occur inherently in that. That's what this is showing you. It's just showing the different combinations. If these are all the sperms and these are all the eggs, you can imagine all the different combinations that can come from that. So let's talk about how this applies to real world. I like this picture because here's dad holding baby, and then here's baby grown up holding new baby. And it's just cool to show how heredity works, I guess. Heredity is a conserved process. What does conserved process mean? This means that this is a process that is something that all organisms on Earth have. We've talked about a couple of these with cellular respiration, photosynthesis. Photosynthesis is not something all organisms have, but like glycolysis would be one, would be a conserved process. And so this was just something that was in the AP literature, and I felt like I needed to mention it. All right, so let's talk about a couple of these terms here. A gene is a section of DNA that codes for a protein. A trait is the physical characteristic exhibited by that organism. So like you have a gene that codes for eye color. Eye color is the trait, right? That trait is determined by a particular protein. And there are different versions of that trait. There may be blue eyes, red eyes. Nobody has red. Well, you get the idea. And typically with alleles, we talk about them as dominant or recessive. Mendel had a law called the law of dominance, and he said that the dominant allele always expresses itself. That the recessive allele can express itself only if there's no dominant present. Mendel was generally correct, but there are some exceptions to this. Allele only has 2 L's. And that brings us to genotype and phenotype. Genotype has to do with the genetic makeup of an organism. Usually you'll see words associated with genotype like homozygous. I think I have that. Maybe I don't. We'll see. Homozygous, heterozygous, homo same, hetero different. Zygous has to do with zygote. And so like a homozygous individual would have the same zygote, right? That they got a big A from mom and dad, or like that. Or heterozygous would be different zygote. One big A and one little a. Phenotype is what you see. The way I remember this is phenotype, and the word physical has a PH in it as well. And so that's how I remember that. Law of dominance. We talked about this already. Dominant always expresses itself. If you look at this picture, which one's dominant? Red. It's dominant. Even why isn't it half red and half yellow? Because dominant always fully expresses itself, unless there's some differences, which we'll talk about. But recessive only expresses itself when dominant is not there. And we talked about the law of segregation already. The law of segregation says that big T and little t have an equal chance of being inherited because they're going to split up. And so this is a 50-50 chance, and this has a 50-50 chance of being inherited as well. And then we talked about independent assortment. We said that we gave the example up above, so you can go back and look at that, but that is also a very important idea in dealing with heredity. And so how is this going to translate to the AP exam? Well, it's going to be some kind of probability problem, more than likely. And so this is the actual thing that's printed on your AP Bio formula sheet. And so I'm going to describe to you how to use this rule, which just seems like a bunch of gobbledygook. I wish I had a blank slide. There's my blank slide. Oh yeah, traditionally you use Punnett squares to do this. Punnett squares are long and arduous, so we're not going to do those unless you just want to. I feel like it may be a waste of your time, particularly in the multiple choice section. So let's talk about how you can do this simpler. There used to be more problems like this on the exam. I haven't seen one, but you never know when you might see one. So let's say we have an individual that is heterozygous for 3 genes. And we have another individual that is heterozygous for 2 genes and homozygous dominant for the third gene. We're not even going to talk about what these are because we're just going to do the probability problems. Well, there's two different ones, right? There's A or B, then you add. If they're mutually exclusive, the or is the important thing here. If it's A and B, then you multiply. And is the important word here. And so how do we do this? Well, let's say, what they're going to ask is they're going to say, here's mom and dad, and what's the probability that you're going to have this individual, right? And you're going to be asked to give the probability for that. So what do? Well, you use those two rules of probability to do it. And so the way I do this is I take each one separately. And so if we were to do this with the Punnett square, I'm going to show you the long way because it may help some of you. So we're going to draw our Punnett square just for the A's. And I know that the chance of getting that big A, little a is 1/4, right? The chance of getting this big A, little a over here is also 1/4. And so the chance of getting that one or that one is 1/4 plus 1/4 because of my rules of probability, right? And so that's 1/2. And so the chance of getting big A, little a is 1/2. This mom or dad, let's go ahead and call this dad. Let's call this mom, just for the sake of conversation. Well, what's the chance of getting big A, little a over here? It's the same, 1/2, right? And so, or of them donating big A, little a, I guess you should say. And so, what is 1/2 times 1/2? Well, why am I doing that? Because it has to be this and that. It has to be, right? And so that is 1/2 times 1/2 or 1/4. And so that's just for A's. So that's 1/4 just for the A's. What about for the B? Well, what's the chance of getting little b when you mix those two together? It's 1/4, right? Now, because I want this and this and this, I have to multiply all the numbers together. And then what's the chance I'm going to get big C, little c with these two C's? It's 1/2. And so 16, so that's 1 out of 32 chance that this child will be born to that individual. All right, hopefully that was helpful. I haven't seen a problem like that on the exam for a little while. Used to when we had exclusive math problems, you saw it every single time, but you never know. And so there's just how you work those problems. All right, let's talk about linked genes. Linked genes has to do with crossing over. It's going to be used in that same context. And basically, a linked gene is genes that are found on the same chromosome. And so in this particular case, G and A are linked genes. And the way that you determine how far G and A are apart is the percentage from which they cross over. And so what is the chance that, well, let's ask it a different way. Is the chance that crossing over is going to happen here or crossing over is going to happen here greater? Well, it's obviously the one on the left, right? Because there's more places that crossing over can occur. And so let's say that crossing over happens 90% of the time here, but only 6% of the time here. And so they're going to take that number and they're going to turn it into something called map distance. That's it. So this is 9% crossing over here, 11% here. So you could add those together and you could say that 20% of the time crossing over happens between B and VG. There used to be a lot of math that was associated with this, and sometimes you'll see it called centimorgans or map units like MU. I don't expect any problems to be with this, but just kind of showing you, that there's some of the things, and this is that centimorgans again. Again, it's just addition. You don't want to, you don't want to try to overcomplicate this. And so this, double crossover. So you could literally waste hours thinking about this kind of stuff, but you might see one question on the exam from it. And so it's one of those things that I would just go ahead and sacrifice this to the AP Bio gods and wouldn't worry myself with it other than just a general understanding of how that works. And I know it maybe sounds like a cop-out, but I just don't understand how this is going to help you. And so I don't think this is going to be feature. I haven't seen it on an exam, and I don't think it's going to be that important. All right, sex-linked traits, very important idea. Sex-linked traits are traits that are found on the X chromosome. Technically, it could be X or Y chromosome, but in all the problems you're going to look at, has to do with the X chromosome. Why is this significant? Because males don't, they only have one X chromosome. Females have two. And so the inheritance pattern is completely different. It's not going to be your standard, like this sort of thing. It's going to be almost every time you're going to see them write it like this. And so if you're doing like a Punnett square, just so you can think about it, you're always going to fill in your X's and Y's first. All right. And so like colorblindness is a sex-linked recessive trait. And let's say that dad is colorblind and mom is a carrier for colorblindness, right? So she's heterozygous. And so what is the probability for their children? So you would literally just come in here and fill in the little A's. Now notice, dad, there's nothing on the Y because that gene's not found on the Y chromosome. And so mom passes that to their sons. Dad only passes the Y chromosome to his sons. And so for a daughter to have colorblindness, both mom and dad have to pass it to him. But for a son to have it, just mom. Dad doesn't give colorblindness because it's not found in the X chromosome. So you'll see some, one of the problems that I've seen crop up a lot in the last couple of years is what kind of inheritance pattern does this demonstrate? And so the way that you would check to see if it's a sex-linked inheritance pattern is it's going to be much more prevalent with males than it is with females. And that's going to be your tip-off there. And this shows like a pedigree for that. Pedigree is just a way of showing the passage of genes in a family, and they're usually, you'll see them labeled like this, right? And this is showing X-linked recessive. So it's X-linked. It's another way of saying it. And the circles are always female. Males are squares, and then the shaded is going to be the particular disease or whatever that's being tracked. And so notice mom has it. And so what does mom give to sons? Only the disease, right? And so the daughters may or may not have it because, well, they don't, obviously. And so dad doesn't have it either. But dad only has one big A here, right? So if you were to do dad's genotype and do like that. And so you could go through and track all of these, right? The dark ones are easy because they have it, but then some of them you may not be able to tell. So like mom is for sure giving daughter one little A, right? But her other A is big A. And so maybe you can't know all of them because, we know that this guy gives that because he doesn't have the disease. But so this daughter right here, number 10, could be that or that. And so there's some things you may not know. And so a question in FRQ may be like, what's the genotype of individual number 10? And, predict and then give your justification. And you could say, I don't know, it could be one of those and justify. Some inheritance patterns are different, like multiple genes. Basically what this means is that you have multiple genes that code for the same phenotype. So skin color is a great example of this. You have multiple genes that code for the same phenotype. There's several genes that code for skin color. For instance, skin color is the phenotype. And what this is showing you is like, it's called like an additive effect. And so basically the genes kind of add up depending on which ones are turned on and off. The way that I like to think about it is like a light room, or let's say you had a room that had 21 light switches in it. And if you turn all of them on, it's all the way light. If you turn all of them off, it's all the way dark. And then any combination therein is going to be completely different. Like if you turn on lights 3, 12, and 16, it's going to look different than if you turn on 4, 7, and 14, right? And so that's the idea, is that idea of additive effects. Non-nuclear DNA. This is DNA that's not found in the nucleus. The inheritance pattern here is interesting because only mothers give their mitochondrial DNA. And so the way to track this would just be is if it's passed from the mother exclusively. So non-nuclear DNA has to do with maternal DNA passed in the mitochondria. Phenotypic plasticity. This has to do with things that are affected by the environment. So this is a phenotype that is passed down, but the differences in the environmental conditions can cause a particular range of effects or plasticity. And so that's what this means. Under 20 degrees, black ears, not white ears. There's several examples of this. Hydrangeas, the pH of the soil. Stomata density has to do with the amount of CO2 available. Average height in the world. What's the difference between 1896 and 1996? Grocery stores, literally. So just the availability of food and healthcare is going to change the average height of people significantly. What are some things that can change this? I think that's where we're at. Yes, mutations can cause an issue with genes. We've talked about this already, so I'm moving on. Non-disjunction. This is something specific to meiosis because non-disjunction, look at the word, junction means where two things join. Disjunction is where they separate. Non-disjunction is a failure to separate. And so non-disjunction is when chromosomes fail to separate. Homologous pairs fail to separate in anaphase 1. Sister chromatids fail to separate in anaphase 2. What's that going to cause? What's going to cause the individual to have more than the number of chromosomes they should have or less? And if it's non-disjunction in meiosis 1, you're going to see this pattern, N+1, N+1, N-1, N-1. If it's non-disjunction in meiosis 2, you're going to see two possible normal alleles, and the other two are going to be N+1, N-1. Here's an example of non-disjunction. Trisomy 21 is this disorder, or what we call Down syndrome. The individual has 3 chromosomes on their 21st pair. There's several examples of this. Here's one for sex-linked chromosomes. So people who have more than one X or more than 2 X's for females, 2 X's for a male. The presence of the Y chromosome is going to determine the male, and the X chromosome is necessary for life. And so just simply someone who has a Y chromosome is not viable, what it says here, but an X chromosome with nothing else is viable, or 2 X chromosomes and a Y. The presence of the Y, again, is going to determine maleness in that regard. Recessive disorders, this is pretty straightforward. They're passed on the recessive allele. And dominant disorders are passed on the dominant allele. An example of this one would be Huntington's disease. This is pretty much it. And so hopefully this was helpful. Let me know in the comments down below if this was helpful for you, or if you have questions, please like this video, subscribe. I'm hoping to post videos for Unit 6, 7, and 8, hoping to get to those. Good luck on the exam on Wednesday. See you in the next one.